Adam. I'm a PhD student at the Center for Neurodevelopmental Disorders, and I will be joining this year's DevNeuro Academy. So to start with, I'll tell you something about what I'm doing right now. So I'm currently in my first year of the 4-year PhD program. So this year I'm doing rotations in 3 different labs. And based on those rotations, I will then choose a lab to stay in for my PhD. So for my first rotation, I worked with zebrafish to study a neurodevelopmental disorder called FOXG1 syndrome. For my second rotation, I then worked with human-induced pluripotent stem cells. So those are basically cells that are taken from human donors, and you basically just take a hair sample and then you culture cells from that hair sample and you reprogram the cells into neurons. So then if, for example, you take a hair sample from someone who has autism, then you can then culture neurons in the lab that have the same genetic mutations as someone who has a neurodevelopmental disorder, such as autism. And then in my third rotation, which is the rotation I'm actually doing right now when I'm filming this video, so this rotation is looking at spina bifida histology. So spina bifida is a disorder where the neural tube doesn't close during development. So babies have a lesion on their back where their spinal cord is because their spine hasn't closed. And this, of course, leads to all sorts of problems. So, for example, the children have problems walking, for example, because it affects their spine. And I'm looking at whether it affects brain development as well. So I'm basically doing post-mortem histologies of fetal tissue to look at what the effect of spina bifida is on brain development. Now, just to give some background about myself, so before this, I did an undergraduate degree in neuroscience and biology. So I basically did, it was a general biology degree as part of a natural sciences degree at the University of Cambridge, but I specialized in my final year to do developmental biology and developmental neuroscience. So I was in the Department of Physiology, Development, Neuroscience, and my final year thesis project was actually looking at human trophoblast stem cell development and differentiation. So trophoblast cells are cells in the are cells that form the placenta, and of course the placenta is really important in pregnancy to nourish the developing fetus. And what's interesting is that placental development is really important for neurodevelopment, and if you have disorders in the formation of the placenta, or if, for example, the mother is sick with a disease, or like, for example, influenza, then this can affect, through the placenta, the development of the baby, which is why studying placental development is also important for understanding neurodevelopment and neurodevelopmental disorders. I'm really excited to share more with you when we do the DevNeuro Academy, and I'm really looking forward to it. So that's everything for me for now.